Good afternoon. I would like to thank OGT for inviting me here to share our data with you on the validation of the CytoSure Constitutional V3 array in prenatal as well as postnatal analysis. First of all, I would like to take you through our history of array CGH in Leuven. So for our postnatal arrays, we started back in 2004 with a homebrew 1 megabase back array. This was quite time-consuming and labor-intensive to print the arrays ourselves and to have our in-house developed analysis tool. So in 2009, we decided to switch to a commercially available platform, and this was the 2x105K array from OGT at that time. Then in 2010, we switched again to a 4x180K array from OGT, and we selected or we preferred at that time a custom design because of ongoing research projects in the lab. And with this custom design, we could have a higher probe density in genes of interest for these research projects. Then for prenatal arrays that we perform in Leuven, it's a different history. So before 2010, we performed the classical cytogenetic techniques like FISH for a rapid aneuploidy screening followed by a conventional karyotype. Then in 2010, we distinguished between fetuses with ultrasound anomalies and other indications. So for fetuses with ultrasound anomalies, we performed the 4x180K array from OGT, and then for other indications, we still performed FISH and conventional karyotyping. In 2013, we had a national consensus in Belgium that we would switch to array CGH for all invasive prenatal samples. So we don't perform conventional karyotyping anymore in a prenatal setting, so only array CGH for invasive samples. And we also had a consensus that the resolution didn't necessarily have to be as high as for our postnatal samples in order to find the balance between maximizing our yield of detection but minimizing the number of variants of unknown significance that we would detect in a prenatal setting. So just to give you a short or a quick look on our similarities and differences between our postnatal and prenatal array setting. So the platform is different. So we use for the postnatal the 4x180K and for the prenatal the 8x60K array. According to numbers, we perform approximately 3,000 postnatal arrays per year and 1,000 invasive prenatal samples per year. This used to be higher, but because of the implementation of the non-invasive prenatal testing in our lab in November 2013, we saw the decrease in the number of invasive prenatal samples. So according to indications for postnatal samples, the main indications are intellectual disability and developmental delay, and the prenatals are all invasive procedures. For the lab technical part, postnatally we perform a labeling of at least 4 hours and a hybridization of at least 48 hours, while prenatally we try to reduce these times as short as possible because these are urgent samples and we have a very short turnaround time for these samples. So we perform a labeling of 2 hours and a hybridization of 16 hours for these samples. According to calling thresholds, it's the same for both platforms. So we have a minimum of 5 we need a minimum of 5 consecutive aberrant probes in order to call an aberration in the software. And the thresholds for calling an aberration are set at 0.36 for a duplication and minus 0.36 for a deletion. For the reporting, postnatally, we report everything that's above 200 kb, independent of the gene content, but we report smaller aberrations if there's a clear association with the clinical phenotype. And for prenatal arrays, we report according to our national consensus guidelines. So I'm not going to go into detail here because this would take me, again, half an hour to explain this. But if you're interested, you can take a look in our publication from 2014 where we published the Belgian approach for our prenatal array guidelines. So why did we want to validate or test the V3 array? Of course, because of the enhanced exon level CNV coverage of these developmental disorder genes. And as was also touched upon by the previous speakers, it's really focused on 502 genes of interest identified both by the DDD project as well as ClinGen. What was also interesting for us then practically is that there was not really a requirement for further investment in equipment or in training of our personnel, because it's just a different slide, but the procedures, they stay the same. We can use the same scanner, so it's a very easy switch that you can make in the lab. And also, for us, it was also good that, especially for the 4x180K array, that we now have an off-the-shelf availability, because with our custom design, it was customized for us. So it was we had to demand for an order, or we had to place an order, and then the batch was processed or produced, and then we received it. While now this V3 array is off-the-shelf available. So I would like to show you how we performed this validation. So for the majority of samples, we performed the prospective validation, and we also performed a retrospective validation for 5 samples. So for the prenatal array, we performed the prospective validation for 41 samples and postnatally for 80 samples. And I would like to show you the comparison in QC metrics and also in the analytical accuracy for this validation. So according to QC metrics, we compared DLR, the derivative log ratio, which is a very important measure. It's the probe-to-probe log ratio noise, and in our lab we have a cutoff of 0.2. So the DLR has to be below 0.2 in order to consider this as a good quality result. And then we also compared signal-to-noise ratio for the red as well as for the green signal. And here we have a cutoff of 30, so this signal-to-noise ratio has to be above 30 in order to have a good quality result. So as you can mention, the QC metrics are quite comparable, with a slight improvement for the V3 array, but they are all above our cutoffs for good quality. So we were very happy with these results. Also for the 4x180K array, we did the same comparison, and also there we had very good QC metrics compared to the V2. So then for the prospective validation of the prenatal arrays, so the 8x60K array, we processed 41 samples, and we had a concordant result in all of these 41 samples. So for 35, we issued a normal report. Sometimes there was a small difference in size in the benign CNVs, but this is logical because of the difference in probe density in specific regions. And we had an aberrant report for 6 of them. 3 of them were trisomies, 2 trisomies 21 and 18, so it's quite clear that both platforms can detect these. The other ones were somewhat smaller, but I would like to focus on the last one. So this was a deletion on 13q12.11, and we detected this deletion with our V2 platform. But we were wondering if this gene was also involved in the deletion, because there were no probes spotted here for this gene. So we took a look at the V3, and we processed this sample on the V3 platform. And indeed, the V3 platform had 4 probes in this region, and we could now accurately call this deletion and be confident that this gene was also involved in this deletion. And this was just of importance for us, because it was detected in a prenatal setting, and in a prenatal setting, our consensus guidelines, they tell us that we cannot report carriership status of autosomal recessive conditions unless the carrier frequency is higher than 1 in 50 and testing is available in Belgium. And one of these autosomal recessive diseases that have this high carrier frequency is or the connexin 26 mutations, and it's known. This is not connexin 26, but this is connexin 30, and a deletion. This is a recurrent connexin 30 deletion, and in combination with another connexin 30 deletion on the other allele, or with a connexin 26 deletion, this can give rise to autosomal recessive deafness. So that's why we decided to report this in a prenatal setting in order to be able to test the connexin 26 gene and to make sure that there was no autosomal recessive deafness present. So for the 4x180K array, we compared 80 samples, and for 77 of them, we had a concordant result, 55 with a normal report and 22 with an aberrant report. And also there we saw sometimes small differences in size or in the number of probes in the reported region. But this had no influence on the report itself. So I would just like to show you some representative examples. So here is a duplication that was detected. Red is always V2 and blue is V3. So it's almost exactly the same between the V2 and the V3 platform according to size and to the number of probes. This is a deletion of the ARCAT region, and here you see that that for the V2 the region is somewhat larger than on the V3 platform, but also this has no consequence for reporting. The deletion is detected, and we will issue the same report independent of the platform used. So, and this was just an interesting sample for us in the lab. It were 2 flanking triplications on the X chromosome with a small normal region in between. So we were just wondering whether also the V3 would be able to detect this as 2 flanking triplications, and also here it performed very good. So besides these 77 concordant results, we had 3 discordant results, but I already would like to mention that these were all very small aberrations, so under our cutoff of 200 kb. But I will guide you through these 3 discordant results. So for one of them, we detected an aberration on the V2, which we did not detect on the V3, and 2 others we did not detect something on the V2 where we detected an aberration on the V3. So this is the first one. So on the V2 platform, we detected a 7.2 kb deletion of 7 probes here in the BBS1 gene, while on the V3 we were not able to detect this as there was only here 1 probe present in this region. Then for this case, we detected a 29 kb deletion in the intragenic in the SPTLC2 gene with the V3 array. It contained 21 probes, while on the V2 array there was only 1 single probe. And if you take a closer look, it's also in the deleted ratio, but just 1 probe is not enough to make a proper calling. So this gene is responsible for an autosomal dominant neuropathy. So it might be of interest if there's a clear association with the clinical phenotype of this patient. So this can give us more yield and more information for this patient. And also here we detected a 4.2 kb deletion in the KIF2A gene with 14 probes on the V3 array, and there were no probes on the V2 array. So also here, this gene is responsible for autosomal dominant cortical dysplasia. So again, if there is a link with the clinical phenotype, then we would report this. Then for the retrospective validation, we did an MLPA V3 comparison. In our lab, in the molecular part, they are performing MLPA for NF1 and BRCA2, so they have samples available there, and we just selected 2 of each to see whether we could also detect these exonic deletions or duplications on the V3 array. So it was for NF1 a deletion of 1 exon and a duplication of a few exons, and for BRCA2 the same situation. So for NF1, the single exon 5 deletion was not called by the software, but this is because of our setting that 5 consecutive probes had to be aberrant in order to have a call by the software. But if you just take a closer look, then you can do the manual calling of this deletion of this single exon. Then for this, it was pretty obvious that the duplication of the 4 or 5 exons was detected by the V3 array. For the BRCA2, there was a deletion of 1 single exon here, exon 19, and it was accurately detected by the V3 array, while here exon 20 showed a normal profile again. And here it was a duplication of 2 exons, exon 19 and 20, and you can mention here that the duplication was accurately detected. So then we also had one interesting case. It was a woman carrying an NDP exon 3 deletion responsible for Norrie disease. And this woman was pregnant, so we were asked whether we could detect this deletion with our array, and we were still using the V2 arrays at that time. So we took a closer look, and we only had 2 probes in the region of this deletion. So we were not confident enough to make a proper call with these 2 probes. So we received the fetal material, and we did an array gene just to do a genome-wide approach. But in addition, we had to do an additional PCR-based approach to really be confident of the calling of this exon 3 deletion. But at that time, we didn't have the V3 available yet. But if we would get the same question now, we can help this woman with only performing array because now on the V3 platform, 9 probes are involved. So with these 9 probes, we feel confident enough to do a proper calling of the deletion. So to conclude, in the postnatal setting, we detected slightly increased QC metrics, but the QC metrics were overall very good. And from what we already mentioned in our validation cohort, we really expect to have a higher yield with this new array and due to the detection of these exonic CNVs. And we will see in the future because we implemented the array now since the beginning of May. And we are finalizing our validation in the prenatal setting, and we are planning to implement this V3 array in July. So in a few months. So with this, I would like to conclude, and I would like to thank all the members of our lab in Leuven and some persons in particular who intensively participated in this work. Thank you.